Okay, so good evening, everyone. Welcome to the week 8 tutorial session or problem solving session of the NPTEL course Genetic Engineering Theory and Application. So I hope all of you have gone through the week 8 lectures. So week 8 lecture is a continuation of the week 7 lecture. So in week 7 we were studying about the various methods of protein purification. So the various chromatographic techniques. Starting from the ion exchange chromatography, then we learned about the uh, hydrophobic interaction chromatography. So mainly these two chromatography techniques we learned in the last session. In this week, the other two important chromatography techniques that were covered were the size exclusion chromatography, also called the gel filtration chromatography, and another is the affinity chromatography. Okay. So, so today we will mainly discuss these two chromatography techniques in detail. So, once you have so a quick recap of what all we have learned in the previous lectures. Like first, we learned about the how we can identify the target gene. So we can screen the cDNA library or we can screen the genomic DNA library. So once we have got the target gene, then what we do, we will clone that target gene in a vector. So for that, what we do a restriction digestion. So we restriction digest for the vector as well as the gene of interest, and then we perform a ligation reaction. So on performing the ligation reaction, we will get a recombinant plasmid. Now this plasmid is inserted into the host cell. So the process is called transformation. If we are inserting the recombinant plasmid in bacterial cell, or if we are inserting it in mammalian cell, it is called transfection. Now, after introducing the recombinant plasmid in the host cell, then we have to screen which host cell has taken up the recombinant plasmid and which host cell has not taken up the recombinant plasmid, or which has taken up a say for example, it has taken up a self-ligated vector. Okay, so we have to screen for that. So we learned about the various screening methods. So these were all so once we have screened and we have obtained um, the host cell, the correct host cell, which is carrying the recombinant plasmid, then we can use that for our downstream processes. Okay, so these are all the upstream processes. Once we have identified the correct host cell, correct recombinant host cell, then what we do, we will go for expression of the gene of interest. So we will go for protein expression. So for protein expression, so we for that we again learned about the various expression vectors. What all must be present in a expression vector? It must have a promoter, it must have a terminator, and for purification of the protein, you can also add a fusion tag. So we all with these things we learned in the earlier classes. So now after we have expressed the protein of interest, then the next step will be to purify the protein, isn't it? Because the when we are expressing a protein in a host cell, then along with a protein of interest, the host cell will also express other proteins. So different proteins. So for example, proteins which will be required by the host cell for growth, for the metabolism or biochemical reactions. So all these different proteins will be expressed. So from all these pool of protein, we need to purify our target protein. Okay. So let's start today's presentation. Okay, so yeah, in between, if you have any doubts or in the previous lectures or anything you couldn't follow, then you can ask me. I will try to answer the question to solve your doubt. Okay. So I hope the screen is visible. Okay, good evening everyone. I am Akash Jyoti Pathak and I am one of the PMRF for this NPTEL course, Genetic Engineering Theory and Application. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So as I was discussing, so in week 8, we mainly learned about the two main chromatography techniques. One is the gel filtration chromatography, or it is also known as the size exclusion chromatography, and another is the affinity chromatography. So basically, bioaffinity chromatography is a type of affinity chromatography. So the main two techniques are the gel filtration and the affinity chromatography. So in the last lecture, we learned about this IEX chromatography. So what is the principle of IEX chromatography? Anyone? So the principle of IEX chromatography was that it was separating protein on the basis of what? Separation of protein on the basis of charge of a protein. So the charge of a protein will again depend on the pH of the buffer we are using and the PI of the protein. So what is the PI of the protein? That will determine what will be the charge of a protein in a particular buffer. So if the pH of the buffer is less than the PI, then the charge on the protein will be what? It will be it will be positively charged, isn't it? And if the pH is greater than PI, then it will be negatively charged. Okay. So if the pH of the buffer is less than the PI of the protein, then it will be. So what happens basically? So if the pH is less than the PI, then you have a lot of protons in the solution. So all your this COO minus group and your NH2 group, all these so this alpha amino group and the alpha carboxyl group, and if you have again if you have some side chains which are having this NH2 group or the COO minus group, then all these will be charged, isn't it? So this CO minus will get converted into COH, NH2 will get converted into NH3 plus. So that is why when the pH of the buffer is less than the PI, your protein will be positively charged. Okay, and if your pH is greater than the PI, then what happens? These carboxyl group, it ionizes. It ionizes and it forms COO minus and NH2 group. So as a result, the net charge of the protein becomes negatively charged. So these are basic biochemistry which you must have learned. Uh, so IEX is separating protein on the basis of PI of on the basis of charge of a protein, and hydrophobic interaction chromatography was separating on the basis of hydrophobicity, and it was following the principle of salting in, salting out, isn't it? So now coming to gel filtration and affinity chromatography. So what is gel filtration chromatography? So when we are separating proteins on the basis of their size, not only on the size, mainly the correct is thing to say is that we are separating protein on the basis of size and shape of a protein. Okay. So not only the size is important for gel filtration chromatography, the shape is also very important. Why is it so? So for example, consider a protein. So say a protein is globular in shape. Okay. And it is having a molecular weight of say 36 kilodalton. And another protein is having the same molecular weight, 36 kilodalton, but its shape is, say for this example, it has some extended intrinsically disordered regions present in the protein, or it can have some unstructured region or some regions which are not properly folded, or it has some unstructured domains which are present on the protein, and it has an extended structure. So in this case, when you will do a size exclusion chromatography, which of these proteins will come first? If you do a size exclusion chromatography, what you get? So on your so any chromatography techniques will have absorbance 280 on the x-axis and the sorry on the y-axis, and the volume or the volume of elution on the x-axis. Volume eluted, or you can also represent it in the form of time eluted, will be on your x-axis. So this is a chromatography profile. So say this is A protein and this is B protein. And if you do a size exclusion chromatography, then which of these proteins will be eluted first? So what will happen is that the one which is having a more extended conformation or which is having this unstructured regions. This will be eluted first compared to the one which is having a globular shape. So you will get so for this B protein, you will get a peak here, but for the A protein, it will be eluted much later. Okay. So we will learn about this principle of gel filtration chromatography, but just to uh, to to start with this, I discussed a concept because not also not only the size is important, but the shape is of the protein or the hydrodynamic radius. Sometimes this is also called the hydrodynamic radius of a protein is very important when we are doing a gel filtration chromatography. Okay. So what is the principle of gel filtration chromatography? So this is the principle is very simple. So basically, if you can consider that, for example, you have prepared tea and you are straining the tea from the tea leaves. So what do you use? You use a sieve, isn't it? You use a sieve and you add the tea, and the tea leaves get stuck on this sieve, and the tea is strained, isn't it? He is trained in the cup. So the similar thing is done in gel filtration chromatography. So every protein has a particular size and a particular shape. Now what happens in this gel filtration chromatography? We use a column, and this column has beads. Okay, so there are different types of column, and different types of column have different sizes of beads. Okay, now these beads have pores inside them. Okay, the beads will have pores. Okay, so there are different types of columns which are commercially available, and each of these columns will have beads, and these beads have pore. Now, depending on the size of the protein, so what will happen if when you have applied, when you have when you have added the protein that you want to purify. So your protein sample will have different types of, so it is an unpurified protein. So it will have different samples, different molecules. Some will have large size, some will have small size. And there can be protein molecules which have sizes in between these. So what will happen is that the protein molecules which are having small size, they will enter into these pores. Okay, so the protein molecules which have small sizes, they will move inside these pores, and they will pass through the pores. And the ones which are very large in size, they cannot move inside these pores. Okay, they will be excluded from the pore, and they will pass. So they will pass in between these the spaces which are present between the beads. So they will pass through the spaces which are present between the beads. And when you are passing the mobile phase, so what will happen? Your the proteins which are having small size, they will move through the pores, while those having large size, they will pass through the spaces which are present between the beads. So as a result, your large size proteins will be eluted first. So the main thing to note over here is that the large size proteins will be eluted first. Large size protein eluted first, and the small size protein is eluted later. Okay. Large size protein eluted first, and the small size protein is eluted later. Why is it so? Because the ones which are entering the pore, they will take a longer time to be eluted, and the ones which are not entering the pores which are present in the beads, they will be eluted earlier, because they will take less time to traverse through the column, and hence they will be eluted faster. While the ones which are having small size, because of their small size, they will enter into the pores, and they will hence take longer time to traverse this entire length and get eluted. Okay, so basically it is the separation of the molecule is on the basis of what size and shape. So remember this, and the principle is what it is exploiting. It is exploiting the molecular sieve properties. Okay, so because why is it you exploiting the molecular sieve properties? Because these resins are having pores of different sizes. Okay, and the stationary phase. So this is your stationary phase. So what is the stationary phase in gel filtration chromatography? So the stationary phase basically contains gel beads. So these are your gel beads, and these gel beads contain pores that span a relatively narrow size range. The pore size is typically determined by the extent of cross-linking between the polymers of the gel material. Okay, so basically your these beads that I was talking, these are basically gel beads. Okay, and these gel beads are formed by chemical reactions. So for it can be polyacrylamide gel, it can be polyacrylamide agarose composite gels, or it can be dextran based gels. So depending on the chemical reaction that has with the by which these gels have been formed, that will determine the pore size of the gel. Okay. So that is and also it will depend on what percentage of that chemical you are using, isn't it? So if you are using a very high percentage of the of polyacrylamide, then it will have smaller pore size. If you are using low concentration, then it will have less pore size. So that will control the cross linking. So what is the cross linking between these monomeric units of these polyacrylamide state? So if an aqueous solution of molecules of various sizes is passed through the column containing such molecular sieves. The molecules that are too large to pass through the pores are excluded from the solvent volume inside the gel beads. The large molecules will therefore traverse the column more rapidly than the small molecules that passes through the pores. So this is the same thing that I was explaining. So the ones that are larger, they will be eluted earlier compared to the ones which are having a smaller size and smaller shape. Okay. So this figure is taken from the book Voigt and Voigt, Biochemistry Classical Textbook. So what you can see over here is this. So here you see this A figure. So in this A figure, this is the bead. This is a depiction, cartoon depiction of the bead or the gel matrix that is present in the stationary phase. So you can see here that this gel matrix has spores inside it, and your sample has two different molecules. So one is having it is having large molecules as well as small molecules. So when you are passing this sample through this stationary phase, then what will happen? The ones which are having small size, they will move inside the pores, and ones which are having larger size, because they are larger size, they cannot enter inside the pore. They will be excluded from the pore, and hence they will be okay. So they will be eluted earlier. So now this is your column, and is your gel matrix or gel bead. Okay, so this is your gel bead, and this gel bead is having gel matrix. So when you are passing the mobile phase, so your solvent, this is your mobile phase. So what is happening when you are passing the mobile phase? The ones which are having larger sizes. So these blue colored molecules, they are having larger sizes. So they are getting eluted faster because they are not entering inside the pores which are present in the gel bead. Okay, but the ones which are having a smaller size, they are traversing the entire length of these pores which are present in this gel bead, and hence they are getting eluted later. So this is how the chromatographic profile would look like. So for you will have two peaks because there are two different molecules. So you will have two peaks. So the first peak will be that of the molecules which are having a higher size or larger size. So the larger size molecules will be eluted earlier, and the ones which are having a smaller size will be eluted later. So these are having the smaller size. So this is the entire principle. So this entire text you can read. So the same thing is written over here. So it is taken from the book Voigt and Voigt. So this gel bead consists of gel matrix that encloses an entire space. So the sample solution is applied at the top of the column. The gel beads So here you have applied the sample, and then the small molecules. What will the small molecules will do? They will penetrate the gel and consequently migrate through the column more slowly than the large molecules that are excluded from the gel. So your large molecules will be excluded from the gel. They will not enter inside this gel bead, while the small molecules will enter the gel beads, and they will consequently they will migrate a greater distance, and hence they will be eluted later. Okay. Okay, so so as I was explaining, the gel bead has um, is made up of a gel matrix, and there are depending on what type of cross-linking agent you are using, that will determine the pore size of the gel. So you can have large pores and you can have small pores, and there can be beads which are having pore sizes which are intermediate between the large pores and the small pores. So you see this figure that there are there are different types of molecules here. So these are the large molecules. So because they are very large, they are not able to enter these pores, while the ones which are of smaller size, they are able to enter inside this pore. And so if you the size exclusion chromatography profile of a sample having these different type of molecule, how will it look like? So which of these will be eluted earlier? So one is having the largest size, isn't it? So one will be eluted earliest. Then followed by it, the third molecule. It is looking more bigger compared to the second molecule. So it will be eluted next. Then the second sample will be eluted, second protein molecule. Then followed by it, this one, this fourth sample will be eluted. Then this seventh will be looted, then fifth will be looted, and then sixth will be looted. So depending on the size, so the larger the size, that will be looted earlier, and the smaller the size, that will be looted the last. Okay. So so the same thing is explained in the lectures as well. So the column is packed with beads containing pores. So these beads basically have pores, and these pores allow the entry of molecules based on their size. So the smallest size is in the inner part of the pore, followed by gradual increasing size and large molecules excluded from. So you can read this. The same thing is written over here. The large molecule and small molecule is getting separated because you see the large molecules are moving faster because they are having to travel less distance, while the ones which are having small size, they are traversing the entire pore size, and as a result, they are getting eluted later. Okay, now what happened is, so there is one concept which you must understand, that what is the volume of the column. So if we consider the total column volume of the gel is Vt. So if we consider that the entire volume of the column is Vt, then Vt will be what? It will be sum of Vg plus Vi plus Vo. So what is Vg? So Vg is the volume which is occupied by your gel. So these are the entire, if you sum up the volume which is occupied by the gel beads, that will be your Vg. What is Vi? So Vi is the volume of the pores. So these gel beads have pores inside them as well, isn't it? So if you add up the volume which is present in the pores inside these gel beads, that will be Vi. And VO is this area, this volume. So the void space. So when you pack the column, there will be some free spaces between these gel beads. So that is called the void volume. VO is called the void volume. Okay. So the volume of your column will be what? Volume of your column VT will be the volume of these gel beads. Okay. Volume of these gel beads plus this volume of these pores, Vi, and the void volume. Okay. Vg is the volume of the gel matrix. Vi is the pore volume, and V naught is the void volume. Okay, so the volume of mobile phase flow to elute a sample or elute a protein sample from a column, not a column from a column. Elute a sample from a column is known as elution volume. So what is elution volume? So the volume of mobile phase that must be passed for a particular eluent or a particular protein molecule to be eluted is called the volume of elution. Okay. So the elution volume is related to the void volume and the distribution coefficient Kd as V equal to V naught plus K Kd Vi. Okay, so this is one of the expression. Kd is what? Kd is distribution coefficient. We learned in the last class that the ratio, it is the ratio of the concentration of a molecule in one phase over the other phase. So it is Kd. Kd is Ve minus Vo by Vi. So V is the elution volume, V naught is the void volume, and Vi is the volume of the pores. So now these molecules will have different KD values. So the ones which are having KD values 1, then what does it mean? So, basically, what does this? So basically, KD is your distribution coefficient. Okay. So the distribution coefficient of a particular analyte between the inner and the outer mobile phase is a function of its molecular size. So, so if the analyte is large and completely excluded from the mobile phase within the particle, then KD will be 0. Whereas if the analyte is sufficiently small to gain complete access to the inner mobile phase, then KD is 1. So basically, what is KD measuring? Your KD is measuring the distribution of an analyte between the inner mobile phase and the outer mobile phase. So inner mobile phase, by inner mobile phase, what do we mean? That the volume of analyte which is present in this pore volume, or VI. That is why VI by VO basically. Analyte between the inner and outer mobile phase. Okay, so remember this, that if your KD is 0, if your KD is 0 means what? If the KD is 0, it means that the molecule is not able to enter this pore volume. Okay, that is why what will happen, this basically what happens is that is what happens is when you have a very large molecule and this molecule is not able to enter inside the pores which is present in the gel bead, then your VE becomes equal to VO. Okay, your VE becomes equal to VO, and as a result, your KD becomes equal to 0. So what happens is when we do a size exclusion chromatography, so every column that you purchase commercially has a void volume. Okay, so there are different types of column. There are Cephaldex column, Cefacryl column, Superdex column. So all these different columns have a particular pore size. Now, depending on the pore size, it will also have all these columns will also have a void volume. So what we do is when we do a size exclusion chromatography, we usually do not do any fractionation, or we do not collect the protein which is getting eluted in the void volume, because in the void volume, so the protein which is coming in the void volume, it means that such molecules are not getting purified, isn't it? So void volume means that it is not moving inside these pores which are present in the gel bead, so it is eluting as it is. So anything which is coming in the void volume is not collected. Okay, so that is why your KD, why is your KD 0 for large molecules? So why is KD equal to 0 for large molecules? Because for large molecules, your volume of elution volumes becomes equal to your void volume. So as your elution volume becomes equal to your void volume, because it is not moving inside, it is not entering inside the pore. That is why the void volume which is there, that will be enough to elute the protein, isn't it? That is why this VE becomes equal to VO and this KD becomes equal to 0. So KD equal to 0 for large proteins and KD equal to 1 for small proteins. Okay, or for small size proteins. Okay, so why your KD becomes equal to 1 for such proteins? Because the small proteins, they will travel this entire VI. So VI is the volume which is present inside the pore. So that will travel this entire volume which is present inside this um, pores. Isn't it? That will travel entire volume which is present inside the pores. That is why your VE minus VO, that becomes equal to VI, and your KD becomes 1. Okay. So analyte, so remember KD equal to 0 for large proteins and KD equal to 1 for small proteins, small size proteins. Okay, remember this. Uh, So it will be completely excluded. KD equal to 0 means that such molecule will be completely excluded if analyte is sufficiently small to gain complete access. So if it is getting complete access, then your KD will be 1. And if they are due to variation in pore size between the individual particles, there is some inner mobile phase that will be available and some that will not be available to analytes of intermediate size. And so such analytes will have values between 0 and 1. Okay. So your analytes. So your analytes will have different KD values. In case of size exclusion value chromatography, your analytes can have values KD equal to 0, KD equal to 1, and KD which is lying between 0 and 1. Okay. So the small molecules. So these molecules will have KD equal to what? It will have KD equal to 1. The large molecules will have KD equal to 0. And your medium sized molecules can have any value between KD between 1 and 0. Okay. So KD equal to V minus VO by VI. Remember this formula. So analyte with KD equal to 0. So that is what I was explaining. So the large size molecules are getting excluded from the pores which are present inside the gel beads. That is why all the protein molecules are coming in your void volume itself. That is why your VE becomes equal to V0. So if your VE becomes equal to V0, then your KD will be 0. And so analyte with KD equal to 1, they will have what? They will have the elution volume will be equal to the void volume plus the VI. Okay. So this V becomes equal to basically if you substitute this value V equal to VO plus VI minus VO. So your this numerator becomes VI, isn't it? And that is why your KD value becomes equal to 1. Okay. So and if there are molecules which are having KD greater than 1, then in the such analytes will adsorb to the column matrix. So it will get adsorbed. It will remain adsorbed to the column matrix. So remember these concepts. So as I was explaining that there are different types of columns, Cephardex, then Cepharos, then there are again Cephacryl, then Superdex. The different types of columns are present. Okay. So and there are particular pore sizes. So G10, G25, G50, G100. So these are actually a measure of the pore sizes. So depending on the pore size, it will have a fractionation range. So what do you understand by fractionation range? Fractionation range means what molecule it will. So, so consider this one. Cephardex G10 has a fractionation range up to 700. What does this statement mean? So it means that any molecule, any molecules which are less than 700 daltons, only those molecules will enter inside the pores. The others which are having molecular weight greater than 700, they will be excluded. Similarly, for Cephardex G25, it has a fractionation range 1000 to 5000. What does it mean? So molecules which are having the molecular weight in this range, only these type of molecules will enter inside the pores which are present in the gel beads. Okay. So molecules, if a protein molecule has a size, for example, it has a size 5500, then that molecule cannot enter inside the pore. It will be excluded and it will be eluted in the void volume. Similarly, if it has a size, if it has a size less than 1000, then also it will be. So it is actually a range 1000 to 5000. Okay. So the one which is having less than 1000, that will also enter inside the port. Okay. So they are having different fractionation range. So when you are selecting the column, so when you want to do a size exclusion chromatography of your protein, then you have to consider various things that what is the molecular weight of your protein, and depending once you know the molecular weight of your protein, then you will go for the different columns that are available. And for when you are selecting the columns, then you have to look at the fractionation range of the column. So you must select a column which is having a fractionation range which is comparable to the molecular weight of the protein. So, say for example, you are using this Hepatex G50 column, then you must use a protein which is having a size in between these molecular weight range, 1500 daltons to 30 kDa. Okay, so you have to use such type of protein. Okay, and also the length of the column will also play an important role because in size exclusion chromatography, the higher the length of column you use, the more efficient will be the separation, isn't it? So there are different sizes of columns that are available: 320 ml column, 120 ml column, 25 ml column. So you have to also check that what type of what size length of column you will use. What length of column you will use, what pore size you will use, what is the fractionation range of the column. All these things you have to consider. So now answer this question: the distribution coefficient Kd when an analyte is completely excluded from the column in gel filtration chromatography. So remember the formula. Kd is what? It is V minus VO by Vi. Okay, so if it is completely excluded, then your VE becomes equal to VO. Okay, VE becomes equal to VO, and as a result, your KD becomes equal to what? This will be equal to zero. Okay, this will be equal to zero. So remember that molecules which are completely excluded will have KD equal to zero. Answer is option A. Okay. Now moving to the next question. So the gel filtration matrix that is suitable for separating molecules up to 700 Dalton. So what was it? So this number actually indicates the pore size. Okay, the smaller this value, the smaller lesser will be its fractionation range. Okay, so this pore size is very less. G10 is having a very less pore size. That is, as a result, it can only separate molecules up to 700 Daltons. Okay, up to 700 Daltons. Answer is actually Separatex G10. If you see this table as well, which was discussed in the lecture. So G10 has a very small pore size. That is why it can only separate molecules up to which are having molecular weight up to 700 Daltons. Okay. Now, elution volume for an analyte with KD equal to zero is what? So just now I explained that anything which is not which is having a very large size will not enter inside the pore. As a result, it will be eluted in the void volume. So what the void volume of the column? So each column has a particular void volume. So say we have this column, the cefacryl 16 by 26 by 600 in our lab. So it has a void volume of 47. So if So the so this so we do not fraction it in this volume. So because in this volume anything that is getting eluted is not purified. It is coming in the it is not entering inside the pore and it is getting eluted in the void volume. So usually the fractionation is started after the void volume. Okay. So that is why V equal to VO. Now in gel filtration chromatography, the radius of gyration depends on what? So basically radius of gyration is a measure of the shape of the molecule. Okay. It is a measure of the shape of the molecule. Radius of gyration will give you the shape of the molecule. That is why answer is shape of the molecule. And gel filtration chromatography, as I was explaining in the very initial when I started explaining gel filtration chromatography, I gave you an example that two proteins may have same sizes. Say for example, both the proteins have same sizes of 35 kilodalton, but one protein will get eluted earlier and the other protein will get eluted later. And why is it so? Because it can happen that this protein has a more globular shape and the other has a more extended shape. So the one which is having a globular shape will be eluted much later compared to the one which is having a more extended shape. Okay. Now answer this question. The following statement is true. Any doubt till here? Is everyone able to follow? So the following statement is true for analytes with KD equal to 1 in gel filtration chromatography. So KD is what? It is again VE minus VO by VI, isn't it? So the when will you have KD equal to 1? So when this term will be equal to VI. So if this term becomes equal to VI, so basically if your VE becomes equal to VO minus VI, okay, or VI minus VO. Then, then this term will become equal to, so if your numerator becomes here, then it will equal to 1. So the ones which are able to, the ones which are completely, which can completely penetrate the pores which are present in the gel bead, they will have a kd equal to 1 because they will traverse the entire volume of the pore sizes. Okay. That is why Kd equal to 1 which molecules will have Kd equal to 1? Kd equal to 1 will be for those molecules which are completely within the pore of the column. So option B, they are completely within the pore of the column. So you remember this lecture slide. Okay, so this thing concept you remember, if V is equal to VO plus VI. So not only it is traversing the void volume, but also it is the how much volume of mobile phase do you have to pass? So you have to pass the entire volume of mobile phase, which is equal to the void volume and the volume which is present in the inside the pore, which is VI. Okay. This entire volume of liquid you have to pass or buffer you have to pass for eluting the protein. That is why V equal to VO plus VI. Okay. So now answer this question. So the column packing, so all these things are have been discussed. We already discussed this. So the column material is allowed to swell in the mobile phase. It is poured into the glass tube and allow the beads to settle. So this column packing is is a very important step because uh, this thing is usually done in the industry. So there are instruments which takes care of what is the void volume, what is the, so it has to be very accurate. It has to be very uniform. So it is poured into the, so basically you have this you have the resin or you have this slurry. This slurry is poured into this the glass columns, and then it is allowed to, the beads are allowed to settle. So there is an entire industry which are preparing such kinds of column. So so in some labs, what is done is column packing is also done manually. But when you are doing column packing, the problem is that you have to be very careful. So usually pre-packed columns are, people mostly buy pre-packed column because in such those pre-packed columns, these problems of having bubbles in your column or these things can be removed. Okay, so the once the matrix is settled to give a column, it can be tested for the presence of air channel and well packing by flowing an analyte with Kd equal to 1. So all these tests are done in the industry, those who are selling these different types of SEC columns. Okay. So you will pass an analyte KD equal to 1, and then you can actually check, isn't it? So if you are passing, if you know that this molecule has a KD equal to 1 in this particular stationary phase, then and you know the void volume of the column, and you know the internal volume of the, or you know the volume of the pores which is present in the beach, and you are passing an analyte which is having a KD equal to 1, then what you can, how will you check, that your column has been packed efficiently, or there is no pores in the, or bubbles which is trapped in the column. So you pass a molecule which is having Kd equal to 1, and if you are getting that analyte when you are passing this amount of volume, that is the volume. Say for example, your void volume is 35 ml and your VI is, say for example, 20 ml. Okay, and after then what, then where will you get your analyte? You will get your analyte at 55 ml. Okay. So you can check that if you have added an analyte which is having Kd equal to 1, and if your column has been packed properly without any air bubble trap, then you will get your analyte at 55 ml. But if you are not getting your analyte at 55 ml, then there is some air bubble, or there is uh, some when you are, the packing has not been done correctly. Okay. So sample is prepared in the mobile phase. So all these things are, so basically when you are doing an SEC, there are three main steps. One is equilibration, So first you have to equilibrate the column with the mobile phase or with the and the mobile. What is the mobile phase? Your mobile phase is basically the buffer in which your protein is present. So first is equilibration, then is sample injection, and the third step is elution. See, we have used an E-ACTA instrument or this FPLC instrument, fast protein liquid chromatography instrument. Then you will see if you have done an SCC, then you will see that there are three main steps here. One is an equilibration step, then is a sample injection step, and the third is the elution step. Okay, so the equilibration step will be what? So in this equilibration step, so if your column volume is say for example 120 ml, then you have to pass 120 ml through that column so that your entire column gets equilibrated with the buffer in which your protein is present. Okay, okay, so you have to pass that particular volume of mobile phase and using that particular flow rate. So the every column has a recommended flow rate that will depend on the type of beads that you have used and the length of the column also. So all these will determine that what will be the flow rate of this particular column. So that the first step is equilibration. Then in this sample injection, what happens? Your sample gets moves from the loop to your column, and in the elution step, your protein starts eluting. So it is so at that particular volume, you will start seeing peaks or the peak of your protein of interest. So that is your elution step. Okay. So yeah, once you do it in the lab, then you will understand better. But these are the three steps. It is equilibration, sample injection. So in the sample injection, your protein moves inside the is passed to the column. To the column from the tubes, it moves to the column, and in the elution step, you will start seeing the you will start getting the peaks of your protein of interest. Okay. And once you have done this, then you do a column regeneration. So every column has a particular recommended storage buffer. So that storage buffer you can use, or when you are not using it, you store it in presence of 20% alcohol so that there is no contamination or no growth of these bacteria and fungus, and your column is maintained. Okay. You can use it for the next time. So now what are the applications of sex? So how can you utilize size exclusion chromatography in your lab? So one is that one is a characterization you can do, isn't it? So what is the size of your protein, or how pure is your protein? That also you can do by size exclusion chromatography. And another thing, as has been discussed in the lecture, is protein folding. So what will happen? So your urea is a what? It is a chaotropic agent. And when you add urea to your protein sample, your protein starts getting denatured. So here you have your protein sample and you have added different concentrations of urea, 2 molar, 4 molar, 8 molar. So with increasing concentration of urea, your protein will be more unfolded. Okay, so as your protein will move from a folded state to an unfolded state, what is happening to the shape of the protein? So at low concentration or when there is no urea, so the native protein is very well folded. So it is very well folded. Okay. It is having a globular shape. Now, once you start adding urea, your urea will break the hydrophobic bonds which are present in the so the hydrophobic interactions which are present, or the non-covalent interactions which are holding your holding the tertiary structure of the protein, that will get disrupted and your protein will start opening up. So once your protein starts opening up, it will become more extended, or this globular shape will get converted into more extended random structure it will attain. So, okay. So, so what will happen? So you have to these four different protein states: first state, second state, third state, fourth state. So the fourth state is the unfolded protein. The first state is your folded protein. Second is partially unfolded, and third is another intermediate unfolded stage. So the one which is unfolded will be eluted the earliest. Okay. As I was explaining, the globular, if a protein is globular, it will be eluted very later. It will be eluted later compared to a protein which is having a more extended shape. Okay. So that is why your fourth peak will be so At the least dilution volume, you will get your fourth peak, followed by your third peak, or the peak corresponding to your third shape, and then the second, and then the first. So the one which is most well folded will be eluted the last, and the one which has completely unfolded. So once it is completely unfolded, it has become more linearized. It has a linear structure, and as a result, it will not be able to enter inside the pores, and it will be eluted much earlier. So at very less elution volume. So for example, say for example, only you are passing 15 ml of mobile phase, and your this unfolded protein gets eluted. Now for the folded protein, you have to pass even more. You have to pass, say for example, 60 ml of mobile phase, and then you will get your native protein. And the intermediate the protein intermediates, or the unfolded protein intermediates, will be eluted in between these two. Okay. Now, another is the protein ligand binding. Okay, so what is protein ligand binding? How can you study protein ligand binding using size exclusion chromatography? So basically, you have your protein. Okay, and you know that this particular, you have done some experiment, and you have found that this particular protein interacts with another protein P2. Okay, or you want to do some enzyme substrate um, reaction. You know the enzyme, and you know that this is the substrate of the enzyme. And so what this enzyme and substrate what will it do? It will form a complex, isn't it? So how can you check that this is the substrate of this enzyme? So you can do a size exclusion chromatography for that. So your when you do a size exclusion chromatography, what will you get? You will first inject the enzyme. Okay, you will first inject the enzyme, and you will get a particular peak for the enzyme. Next, you inject the substrate, and you will get a particular peak for the substrate. Now you have set up a small reaction where you have incubated the enzyme with the substrate. Okay. Now, then you have after setting up this reaction, the enzyme and substrate, or then you inject it inside the column. You are injected in the column. Then what will you have? You will have a size which is intermediate between the two. Okay, you will have this another peak. So what does it indicate? It indicates that this this enzyme forms a complex with this substrate. So that is why, or or this enzyme plus substrate will have a. So your enzyme will has your enzyme has a particular, or let it be enzyme. Forget the example of enzyme and substrate, because if you do an enzyme substrate reaction, then you will also have a product, and then you will also get a peak for the product. But just consider there are two proteins, and you want to study that if these two proteins interact with each other, then you can also do a size exclusion chromatography. So for P1, you will have a particular peak. For P2, you will have a particular peak. And when you incubate these two proteins together, then there will be a shift in the peak. Okay, so as the size will increase, there will be a you will get the peak much earlier than the peak of the P1 or P2. Okay, so here also, if you see this figure, then also the same thing is happening over here. So there is a ligand which is small, and this ligand interacts with the protein, and you get a ligand protein complex. So your ligand, because it is small, it is eluted much later. So you have a peak over here. Now, now what will happen as you are adding protein? So you have set up a reaction in such a way that you are adding increasing concentration of protein gradually. So gradually, as you will increase the concentration of the protein, your protein will start to form a complex with the ligand. And as a result, what you will see that you will start seeing a peak, getting a peak which will have a higher molecular weight than the ligand and the protein, and hence you will start seeing a peak which is appearing earlier than the peak of your ligand. Okay. So that is what has been explained here. You can read, as the concentration of ligand will increase, what will happen? This ligand protein complex will start to form, and then you will start observing the peak, which will come much earlier than the peak of the ligand. And so you can do such kind of studies as well. Now, protein desalting. So what is protein desalting? Is basically your protein is in a particular buffer. Say, for example, your protein is present in in 300 millimolar KCl salt buffer. Okay, KCl 10 millimolar tris buffer. Okay, now you want to remove this KCl from the protein. So there are different types of desalting columns which are available. So what is done in this desalting columns is that these D columns are packed, okay, and they also have a particular pore size. Now, once you add the protein, which is present in this buffer, 300 millimolar KCl buffer, what will happen? This protein will pass through through the pore, and it will get, because it is having much larger size, your protein. If you compare the size of a protein and the size of the your potassium chloride salt, okay, so this potassium and chloride, the size will be much less. Okay, so it will traverse through the entire the entire length of the pores which are present on the beads, and hence it will take much larger time. But your protein, because it is much larger compared to your salt, your protein will be eluted earlier. Your protein will be eluted earlier, and your protein will be free from the salt. Okay, your protein will be free from the salt. Now you have, so if you want to remove the salt from the protein, say, for example, you want to exchange, do a buffer exchange. So for buffer exchange also, we can do protein desalting. Okay, so these are things which are usually commonly done in any this if you are working with any protein and you are want to do any characterization or any studies with the protein, you will use this type of techniques. Okay. So you can remove the protein. So you see here the salt, because it is much smaller in size, it is eluting much later compared to the protein. So when you are doing a fractionation, you can collect the protein separately, and then you can dissolve it in your required buffer. So this is a excellent example of buffer exchange also. So affinity chromatography is what? So affinity chromatography. So till now we understood the IEX, ion exchange chromatography. We learned about the hydrophobic interaction chromatography. We learned about the size exclusion chromatography. So size exclusion chromatography is separating proteins on the basis of size and shape. And affinity chromatography, what will it do? It will separate proteins on the basis of their, so it is separation on the basis of the biochemical properties of the protein. Okay. So you can utilize the biochemical property. So for example, if it is a antigen antibody reaction. You have want to purify an antigen, and you know that the antigen which you want to purify has an affinity for a particular antibody. So then what you can use? You can use a column. You can use a column, and in this column, in the resins which are there, you have immobilized the antibody. You have immobilized the antibody on the resin. Okay, so we learned about this oligo DT primer and how you can isolate the mRNA. So we learned about this, isn't it? Isolate mRNA from rRNA and tRNA. So that is also an example of affinity chromatography. So what we were you doing, we know that this mRNA has a poly A tail. It has a poly A tail. So if we attach oligo-DT primer, or if we pass it through column which are having oligo-DT primer, then what will it happen? Well, then this mRNA will get immobilized on the column which is having this which is having this oligo-DT nucleotides immobilized on the resin. Okay, and then uh, and all the other RNA molecules will get washed off, but your mRNA will remain bound to your beads, and then you can elute it by increasing the salt concentration. Okay, here also the same thing is done. You want to isolate, you have a lot of antigen, you want to isolate a particular type of antigen, you immobilize, you use a column which is having the resins which is immobilized with its particular antibody, and then you pass it through the column. The antigen will bind to the antibody, and all other antigens which is not having affinity for this antibody will be eluted uh, eluted earlier, while the ones which is having affinity, they will remain bound to the column. Okay. So many proteins can bind specific molecules tightly, but non-covalently. So here you must understand. So non-covalent interaction, utilizing a non-covalent interaction, your ligand, your analyte is binding to your specific molecule, and then you can Do you have any doubt till now? Anything that you couldn't understand? Because I've been speaking since the last 1 hour and okay. So if you have any doubt, you can ask me straight away. Okay. So in this technique, what is done is a molecule, a ligand that specifically binds to the protein of interest. A non-reactive analog of an enzyme substrate is covalently attached to an inert matrix. So basically, yeah, in size exclusion chromatography, we were utilizing the pore size of the beads. Okay, the gel beads that we were using. Here, what we are doing, we have beads, and these beads are immobilized with a particular molecule. It can be an antibody molecule. It can be it can be a substrate also. It can be an enzyme also or a substrate also. You can directly immobilize the substrate and then you pass the enzyme. You are having different population of protein molecules and so we will learn one by one. So you have GST column purification, you have nickel NT affinity chromatography. So we will understand, we will discuss each one of them. So it can be anything, anything. It can be an antibody immobilized on the resin. It can be a substrate molecule immobilized on the resin. It can be a metal ion which is immobilized on the resin. Okay, so the target molecule can form different types of non-covalent bonds with these proteins, and then subsequently it can be eluted by say, for example, if it is an enzyme substrate reaction, then we can do some competitive reaction. We add the substrate in more amount or we add the enzyme in more amount so that your so we will learn this. Okay. So when we will study about GST chromatography, I will tell you how this enzyme substrate reaction can be utilized for purification of a protein. Okay, so when an impure protein solution is passed through this chromatographic material, the desired protein will bind to the immobilized ligand. Okay, whereas other substances are washed off through the column with the buffer, the desired protein can then be recovered in highly purified form by changing the elution condition to release the protein from the matrix. So different types of this. Actually there are different types of affinity chromatography. So when we will learn each one of them, then you will understand because but the basic principle is that you will it is following the this affinity means your molecule or protein of interest will have some affinity to the column because of its properties or because of the property of the protein, and as a result it will bind to your column utilizing some biochemical properties. It can be an enzyme binding to your substrate, it can be an antigen binding to the antibody, and then we can change the mobile phase accordingly, or we can use a you use a more amount of substrate and elute the protein, or we can utilize add a competitive ligand or a counter ion so that your protein gets eluted. Okay, so the opera is therefore often greater than that of chromatographic techniques. So here, one thing to note is that this, if you are doing an affinity chromatography, your affinity chromatography has a much higher capacity to purify protein than other types of chromatography. Okay, why is it so? Because here in affinity chromatography, you are utilizing the protein's unique biochemical properties. So every protein has a unique biochemical property. So that is what you are utilizing here. But in other cases, you are utilizing the physico-chemical property of the protein, isn't it? So the size of the protein you are utilizing. So there can be proteins which are having the same size as that of your protein. So that is why. But the biochemical property of a protein is very unique. So that is why the separation of an affinity chromatography is comparatively more efficient than the other chromatography techniques. So you have different types of affinity chromatography. This is from the lecture slides. So It can be a bioaffinity chromatography, it can be a pseudo-affinity chromatography, it can be a dye-affinity chromatography. So what is a bioaffinity chromatography? So it is another type of affinity chromatography where biomolecules are used as receptor present on the matrix. So you have your matrix and your matrix has been immobilized with a particular type of receptor. And then the receptor will be exploited. So this receptor will have affinity for a particular type of ligand. So utilizing this biochemical property of your ligand, you are purifying it. Okay. Antigen antibody. In addition, enzyme substrate or enzyme inhibitor is also an example of this class. So example is GST glutathione. So GST is basically glutathione S transferase, or it is an enzyme, and the substrate for this enzyme is glutathione. So this GST based purification is very regularly done in different labs. So these things you must know if you are, so these are some basic concepts which you must understand. Okay, and so bioaffinity is basically using a receptor, or it can be an enzyme. So you are using any biomolecule. Okay, so you're using biomolecule immobilized on the resin for separation of your protein of interest. So that is bioaffinity. If you are using biomolecule immobilized on the matrix for separation of a protein. Then what is pseudo-affinity chromatography? In this affinity chromatography, a non-biological molecule is used as a receptor on matrix to exploit the separation and purification of biomolecule. So non-biological molecule is used as a receptor. Is used as a receptor on a matrix. So it can be. So here you can also consider the example of nickel affinity chromatography, nickel NTA. So that is nickel NTA is a non-biological molecule. It is a metal ion. Okay. Then there can be dye affinity chromatography. So in this method, matrix is coupled to the reactive dye, and the matrix bound dye has specificity towards a particular example. For example, you have this Sibacron blue F3GA dye coupled to Desktop matrix, and this particular dye has strong affinity towards dehydrogenases. Then there are metal affinity chromatography, or it is also called as IMAC chromatography, immobilized metal affinity chromatography, and this is also very regularly done in labs which are working on proteins, or you are want to express and purify your recombinant protein. Then this nickel NTA chromatography or cobalt NTA chromatography, zinc NTA chromatography. So basically, what is done? You have this column which is present. Your column has beads, and these beads have this NTA molecules, NTA, which is bound to this nickel ion. So this nickel ion can form coordinate bonds with our histidine tag protein. Okay. And so we will also discuss this nickel anti-affinity chromatography in the next upcoming slide. So it will form the coordinate bond with the recombinant proteins which has this six histidine tags fused to it. Okay. And then you can add imidazole, and then once you add imidazole, so this imidazole, because this histidine residue, amino acid residue, if you see this histidine also has a it also has a chemical group which is similar to the structure of the imidazole. Okay, it also has imidazole group. So there is competition between this histidine and this imidazole, and as a result, your imidazole, once if you add more a very high concentration of imidazole, what will happen? Your protein will no longer remain bound to your column. Rather, your imidazole will bind to your nickel ion, and your histidine tag protein will get eluted. Then another is the covalent chromatography. So this is type of chromatography. So here what you are utilizing the thiol group. So you see this figure basically. So you have an activated thiol group. So your thiol group is basically this SH. Okay. So this is SSR, and you have HS protein. If you read this, so where binding of an aniline to the matrix is not reversible. So this is a covalent chromatography. So it involves the formation of a covalent bond between the functional group present on the matrix and the analyte. So the thiol group SH present on neighboring residues of proteins from disulfide bonds after oxidation and under reducing environment. So your SH, basically this SH in the reduced form, it will exist as SH. So your cysteine group, it exists as SH in the reduced form, but in the oxidized form, this hydrogen will get oxidized, and you will have this disulfide bond that will be formed. Okay, so the matrix in the covalent chromatography has immobilized thio groups. So immobilized thio groups means what? It will have this SH groups. Matrix will have SH groups, and it will form covalent linkage with the free thiol group containing protein present in the matrix. So in the oxidizing environment, what will happen in the oxidizing environment? Your thiol group, which is present on the protein which is having cysteine. So your cysteine will have thiol group, isn't it? So in this oxidizing environment, this hydrogen will get reduced, or it will get oxidized. It will be removed. Hydrogen will be removed, and this SS will form a disulfide bond. So this cysteine carrying amino acid protein, and this one which the matrix which is having this thiol group, they will start form a disulfide bond. And then you can do a washing step to remove the non-specifically bound protein, and then you can add reducing agent. So once you add this reducing agent, what will happen? Your SS will get reduced to SH, and your protein will get eluted. Okay. So once you add this reducing agent, RSH in excess, your this one SS that has formed. SS is your protein is having cysteine residue. That is why it has formed a covalent bond with the matrix of the bead, which is also having this thiol group. So this will get reduced, and it will form SH, and your protein will get, you see, your protein is getting eluted as HSP. Okay. So there are different types of affinity chromatography, and yeah, there can be different types of molecules that you can use. So if it is a histidine tag, if your protein has a histidine tag, you will use a nickel NTA chromatography. If your protein has a GST tag, you will use reduced glutathione. If your protein has biotin group, you will use avidin, streptavidin. If your protein has if your protein has this one, if it is a immunoglobulin that you are, if you are wanting, if you are trying to separate or purify your therapeutic antibody protein, then you will, what type of column you will use? You will use this protein A, protein G column. So these are protein A, protein G is agarose Bs which are having these different A and G. So you can read about this, how this protein A and protein G specifically binds to immunoglobulin. Okay. So if it is a glycoprotein, then glycoprotein has affinity for lectin. If it is an RRN, it will have affinity for lysine. If it is a dehydrogenase, it will have affinity for AMP. So different receptors you can use. So you remember some of these, okay? Especially this GST and nickel NT, you remember. For GST, you use glutathione, and for nickel NT, you use this histidine tag protein, 6S histidine tag protein. Okay. So again, this figure is taken from this Voigt and Voigt biochemistry book. So here you can see that in the, this is the nickel NT column, and this nickel NT column have beads. So these beads have a particular shape, or it has a particular affinity for your target protein of interest. So if it is an enzyme, it will have a affinity for its substrate. So it will only bind to your substrate, while the other molecules will get eluted. Okay. So here also you will have this different step. There will be an equilibration step, then there will be binding. There will be basically equilibration, then there will be sample injection step, then there will be column wash, and then you can do a gradient dilution or column wash or elution. Okay. So there will be binding, and then there will be elution. So in immunoaffinity chromatography, what will you use? Your receptor will be your antibody, basically. Your receptor will be antibody. But in this nickel in metal chelate affinity chromatography, your like this one will be your matrix. Your matrix will have this divalent ions. This basically these D group elements, zinc, cobalt, nickel, these can be utilized because it will form bond with this histidine tag. Okay. The same thing is written here over here. So either how will you elute that? So once it is bound, how will you elute your protein of interest? So in the elution step, what you can do, so you can elute it by washing the column with a solution containing a high concentration of free ligand or a solution of different pH or ionic strength. So basically, when we will learn about this GST column, what is done? So you add the substrate of this GST. So you add glutathione in increasing concentration. So once you add glutathione in increasing concentration, then what will happen? Your GST tag protein will no longer remain bound to the immobilized glutathione. Rather, it will bind to your free reduced glutathione and will get eluted. Okay. So here you see this image is taken from this website, cubebiotech.com. So here you can see that this is your recombinant protein. So you can express a recombinant protein having 6 hex histidine tags. So it has 6 histidine tags. So these are your histidine residues. So you can add this histidine tag in the N-terminal part of your protein or your C-terminal part of your protein. So accordingly, so there are consecutively 6 histidine residues which is tagged to your protein. And your this histidine tag forms coordinate bond. So this is your resin. So basically, this is your resin. So your resin and your resin is immobilized with this NTA. So this is nitro. Um, This is nitrile I don't know, forgetting the name. NT is nitrile triacetic acid. Some uh, NT is what? Nitrilo triacetic acid. Yes. NT is nitrilo triacetic acid. So this has this particular structure. CH2, this COO, CH2CO. So this structure is present. This one. So this forms a coordinate bond with the nickel, and then this nickel forms this coordinate bond with your histidine tagged protein. So there is a coordinate bond between the histidine tag. So it is forming 6 different coordinates bonds. So it is forming 2 coordinate bond with the histidine residue, and 4 other coordinate bonds are found with the NTA molecule, nitrilo triacetic acid. Nitrilo triacetic acid. So acetic acid is the CH2COO minus, and you have this N. So there are 3 CH2COO minus groups. So one is this one, CH2CO minus, another is this one, CH2. This one is another, and then there is another. So there are these 3 CH2CO minus groups. And these 3 CH2CO minus groups form this coordinate bonds. Basically, this O minus group and one of this N forms 4 coordinate bonds, and the 2 coordinate bonds are formed with the histidine residues. Okay, so remember this. Sometimes these things are asked. Okay. So here also you can see this figure. So this is your resin, and this resin has been immobilized with this NTA molecule. NTA molecule. So it is nitrilo triacetic acid. So N, then they have CH2CO minus, again NCH2CO minus. So there is 3 acetic acid. So try this is acidic, three acetic acid molecules you have, that is why it is nitrilo. So this nitrilo triacetic acid, and this nickel is forming covalent forming coordinate bond with these oxygen ions O minus O minus. So there are it is forming 3 coordinate bonds with O minus, 1 coordinate bond with this nitrogen, and 2 coordinate bonds with the histidine residues, with 2 of the histidine residues. Okay. Okay, so remember this figure. You will understand. So affinity chromatography, what it is utilizing non-covalent interaction. Non-covalent interaction between your protein and the matrix. Matrix protein. Okay. So this is your ligand. So your ligand can form different types of non-covalent interaction. It can form salt bridge. Salt bridge means electrostatic interaction. It can form hydrogen bonding. It can form van der Waals interaction. It can form pi-pi interaction. So these different non-covalent interactions can exist between your receptor and your ligand. And once it is bound, then you can accordingly use a counter ion, or you can use increasing concentration of a competitive molecule which will compete with your ligand and will elute your molecule of interest. Okay, so this will follow this one. This Kd is equal to RL by, because what is forming this receptor and ligand is forming a complex. So the affinity, it is the, that is why this affinity Kd is what is dissociation constant. It is a ratio of the free receptor molecule, free ligand divided by the receptor ligand complex, concentration of the receptor ligand complex. Okay, so now how will you generate the receptor? So here again this generation of receptor has been explained. So for affinity chromatography, you need to generate the receptor. Okay. So there can be different receptors. There can be a immunoaffinity receptor. There can be a metal affinity receptor. So how do you generate these different receptors? So it can be an antibody as well. So how will you generate the antibody? So the receptor molecule present on the on the matrix can be produced by genetic engineering or isolation from the crude extract, or in the case of antibody, it is produced in mouse rabbit model and purified. So you can either use genetic engineering, you can either isolate from crude extract, or you can generate an antibody. Okay, so we will learn how an antibody is generated in the next slide. So how do you generate an antibody? So this is a very basic principle, isn't it? We all know how antibodies are generated in the body. So in layman's language, yeah, once an antigen infects our body, that antigen will trigger the immune system of our body, and once the immune system is triggered, our immune cells, these B cells and B cells will start producing antibody, and after several rounds, what we will get an antibody which is very specific to the antigen, and that antigen will be neutralized. Okay, that is the principle of antigen antibody reaction. So how can you generate an antibody? So if you want to generate an antibody against a particular protein, what will you do? You will take that particular protein and you will inject it in, either you can inject in mice, you can inject in rabbit, you can inject in monkey, and you can inject in goat, so you can have anti goat antibody, anti mouse antibody. So you have different types of antibodies that you use for Western blotting, isn't it? So the same thing is done. You take an antigen, you do an immunogen preparation, so you cannot directly inject the antigen. You need to add some. You either have to modify it so it doesn't get degraded, or you have to add some other chemical moieties to it. You can add an adjuvant that will trigger the immune system um, even uh, more activation of the immune system. Okay. Then once you have the immunogen, you will inject it in the in a model organism, and in a host organism, and then yeah, then you can collect the serum, and from the serum you will get the antibody, and then you can test that that what is the affinity of that antibody for your antigen. So you have to do a screening test, and then you can purify the antibody. Okay, so how do you generate it? So you have to prepare the antigen, then yeah, it is required for multiple injections to induce robust immune response. So like when if you remember during COVID, we were given several booster doses. So first dose, second dose, third dose. So so in a similar way, here also you need to inject the antigen in several doses at specific intervals of time, so that the antibody is generated in even larger amounts, and then you can purify the antibody. Or you can directly produce the antigen by recombinant DNA technology. You have the antigen sequence, you clone it, and then you express it and purify it. Or you can isolate the antigen from E. coli. So either you purification of antigen under native condition, so you can purify it. So if you have recombinantly expressed the antigen, then you purify it. Or you can either, if your antigen, you can either purify it in native condition. That will again depend on the antigen. So if your antigen is soluble, it can be purified in native condition. If it is insoluble, you can use denaturing conditions. Now, how can you prepare the immunogen? So once you have the antigen, you will, your antigen is mixed with this one. So remember this Freund's incomplete adjuvant. So basically this Freund's incomplete adjuvant. So adjuvant is any molecule which will trigger the immune response of the host organism. Okay. And also another thing is that it will stay in the body for a longer time. It will not get eliminated. So the immune response is increased. So to a final, so you mix this antigen with your adjuvant, and then you get an immunogen. So mix thoroughly to obtain a immune immersion using a syringe or pipette. So repeated booster injections has to be applied 4 to 5 times after every 4 weeks to generate a robust immune response and development of memory B cells. Okay, so once you have the memory B cells, then that can be triggered to synthesis to generate your antibody. Now, in vivo immunization of rabbit. So before immunization, you take out zero point 0.1 to zero point 0.5 ml mice blood from the tail vein before the first injection. Incubate the sample at 4 degrees Celsius at 30 minutes and allow the blood to clot. Centrifuge the samples at 7000 G for 10 minutes. Collect the serum and store it at minus. So basically, you take out the blood. So once you take out the blood, then your blood has your serum and your plasma. So when you keep it for some time, your serum and everything, they will get coagulated. Okay, and so all your these protein components that are present, this fibrinogen and all these factors, different factors which are present, they will get coagulated, and you can isolate the supernatant fraction, and your supernatant fraction will have your antibody. Then you take the mice, it can be Balbc strain of mice, and then you inject this immunogen inside this rabbit. Okay, so these are the steps which you these so actually these things are something which you do. When you do, you will understand. So there is actually no then you need to so just understand the principle. You have prepared an immunogen, you antigen, you can either recombinantly express it or you can isolate the antigen from the organism, and then you have to inject it in a mice or in a rabbit or in a goat, and then you will get antibody. So once you get the antibody, you have to screen it, that which antibody is having more affinity, and then you can purify it. So that is the main crux of everything. Okay, so you have to determine. So how will you screen for you? You can do an ELISA. Okay, ELISA is what? Enzyme linked immunosorbent assay. So here you have a plate, you have a 96 well plate, and on the 96 well plate, what you do, you coat the well with the antigen, and then you apply this antibody titer. So again, so once you have done this antibody titer, your one which is having high affinity for this antigen will go and bind to it, and then you do a couple of washes so that this non-specific binding is removed. Then you use another antibody against the primary antibody, and then you add a substrate, and you get a signal. You get a signal, or you can calculate the absorbance. So the one which is showing a higher absorbance would indicate that it is having more affinity for your antigen. Okay. So coupling of the receptor. So once you have got the receptor, so for affinity chromatography, first of all, what you need for affinity chromatography, you need a receptor. So your receptor can be anything. We discussed all the different receptors that are present in an affinity chromatography. You need a receptor. The next step could be what? So once you have the receptor, you have to immobilize the receptor on the matrix. Immobilize the receptor on the matrix. So how will you immobilize the receptor on the matrix? So first, for affinity chromatography, first of all, you need a receptor. So your receptor can be an antibody. Your receptor can be Can be other, can be an enzyme, it can be a metal, iron. Okay, so there can be different substances. Then once you have the receptor, the next thing that we would need, that you need the receptor to be immobilized on the matrix. So how will you do that? Coupling of the, so that is what is then it was explained in the lecture, that how do you couple a receptor on the matrix. So it can be coupled to the matrix by the following step. It can be matrix activation, covalent coupling, deactivation of the remaining. So there are three steps actually. First, you will need to activate the matrix. So once you have activated the matrix, the next step is covalently coupling the reactive group on the ligand. So once you have activated the matrix, there will be some chemical groups on the matrix that will allow this ligand to form, or this receptor to form a complex with the matrix. And then you will add another some other chemical that will deactivate the remaining active groups on the matrix. Okay, so there are different methods. So was discovered. I'm not explaining all these. Okay. So one is this epichlorohydrin mediated receptor coupling. So basically this is your matrix. So this is your polysaccharide matrix, and to this polysaccharide matrix you want to add your receptor. So this is your receptor. So your receptor can be an antibody, it can be any other protein. Okay. It can be an enzyme, it can be a substrate, it can be anything. Okay, so you want to immobilize the purpose is that you want to immobilize the receptor on this matrix. So how do you immobilize this receptor on the matrix? So first of all, you need to activate the matrix. So how do you activate the matrix? You treat it with a chemical, epichlorohydrin. And this epichlorohydrin, so this is for this forms a ether linkage with the polysaccharide matrix. This you see, so this matrix has been formed, and then you add the receptor. So once you add this receptor, because this receptor is basically what it is, a protein, and your protein will have free amine groups. So because your protein has free amine groups, it can form a linkage. So this ether bond that you see, that gets broken, cleaved, and your this CH2NH bond, this H2O leaves as the living group. It will leave this H2O molecule, and this CH2 will form an amine linkage with the receptor molecule, and then you can add your ligand. So your ligand will bind to your receptor. Okay. So in this way, you couple or you immobilize your receptor on the matrix. So so remember these things. So epichlorohydrin activates the polysaccharide matrix. So your matrix is what? They are all polysaccharide materials. It is either polyacrylamide based, it is dextran based. So they are all polysaccharides basically. They are all polysaccharide. So this activates the polysaccharide matrix. It will by adding this oxidine groups. So this is your oxidine groups. If you remember little bit ether and this organic chemistry, will know that this is this oxidine group with a 3 carbon alcohol group space around. Now activated matrix will react with the receptor. So your receptor has primary amine. Why? Because your receptor is a protein molecule. It will have free primary amine group. N terminal will have free primary amine group, or it can have any other side chains, amino acid residues with side chains having this amine group. And then this activated matrix will react with the receptor containing primary. Yes, receptor will couple to the matrix via thioester or secondary amine linkage. And so there can be either this is the ether linkage that has formed. So you can also amine linkage can also be formed. So this one, this if you see this linkage, this is an amine linkage. So other than a amine linkage, there can be a thiol linkage as well. So thiol linkage will have what? It will have S and receptor. It can be a thiol linkage, it can be an amine linkage, or it can be a ether linkage. Ether linkage will have O receptor. So these are different. So these are this is another branch of study where people do different type of chemical reaction, and you need to this is linkage chemistry you can do to immobilize your receptor on the matrix. Okay, ether linkage, amine linkage. So different reactions you can do, and you can get these type of immobilized matrices. Okay, then another is this carbodiimide mediated receptor coupling. So this is how a carbodiimide molecule looks like. So your COOH will react with this carbodiimide, and then it will form this COOCNH NHR, and then this R This is actually isourea ester. So this isourea ester will then react with the free amine group which is present on the receptor, and then you will have this one amine linkage with the matrix. Okay. So remember these things. There is nothing to understand here. So you have this, you have carbodiimide and it forms a linkage using isourea ester. You will get isourea ester and then this will again react. So there are three main steps. First, there is the matrix activation. So this is the matrix activation step. So matrix activation step is what you are treating the matrix with a particular chemical. So this is your matrix activation step. And then after that, what you are doing? You are adding the receptor. In the next step, what you are doing, you are adding the receptor. Your receptor will go and attach to your matrix, and then you will do matrix inactivation. So matrix inactivation, so that the other molecules get inactivated, and only your. So okay, so now the method used for receptor coupling to the matrix in affinity chromatography is what? Which method is used? So as now we learned for receptor coupling, we can use epichlorohydrin based receptor coupling, or we can do this carbodiimide based receptor coupling. So remember these things, okay? So there is no, so you have to remember there is nothing else you can do about these things because coupling. So these are following the principles of different organic chemistry reactions. These different uh, immobilization is done. Okay. Option is A. So basically you have this matrix. So your matrix, this is actually epichlorohydrin. So it has Cl, CH2, CHOH, CH2Cl. So basically it is CH2, CH, CH2Cl. So this is ClCl and then this is you have OH. So this will react and this will form an oxidane molecule. Oxidane, this is CH2, CH, and you will have an O group over here. So between these two. And then this will go and interact with our matrix and it will form a activated matrix. So once you have the activated matrix, that activated matrix can react with your receptor and that will get immobilized on the matrix. Okay. So you can have different linkages, as I was saying. This is an amine linkage. This is a thiol linkage. This is an ether linkage. The same thing has been explained here. Reacts with hydroxyl group on polysaccharide matrices. So this polysaccharide matrices has free hydroxyl group, and to the this hydroxyl group, this molecule will go and react, and then you will get this molecule. Okay. Okay, the following method is following is a method to elute analytes in affinity chromatography. So how do you elute analytes in affinity chromatography? So basically you increase the concentration of a counter ligand. Okay. So the answer is increasing the concentration of a counter ligand. So as we will see in the nickel NTA, we saw that what is nickel NTA doing? This nickel molecule is forming coordinate bond with your histidine molecules, isn't it? So this was your histidine ring. Histidine also has this imidazole rings. Okay, so it was forming covalent bond with this. But when you increase the concentration of free imidazole, so you are increasing the concentration of free imidazole, then what will happen? Then your nickel will form covalent bond with the free imidazole, and it will because these are non-covalent interactions, isn't it? So the more it will find if the free imidazole is in more concentration, it will start forming bonds with the free imidazole rather than forming bonds with the histidine tagged protein molecule. Okay, so can we read all these things? So affinity chromatography counter ligand is introduced into the elution buffer, which competes with the target analyte or binding sites on the immobilized ligand. So this is the same thing. The following is mixed with antigen to prepare the immunogen. So what do you mix with the immunogen? With the antigen. So you mix an adjuvant. You mix an adjuvant with the antigen to get an immunogen. So what was the name of that adjuvant? It was Freon's incomplete adjuvant. So it has a particular chemical composition. Not going into that detail, but there are molecules which will increase the shelf life or it will increase the ability of this antigen to trigger the immune system even higher. Okay, so this is Freon's incomplete adjuvant is used to enhance the immune system to an antigen when preparing an immunogen. So create the immunogen. So if you apply any antigen, that will not create an immune response. So if it is a carbohydrate molecule, then it does not trigger the immune system. So you need to attach some adjuvant to it so that the immune system can be triggered and the antibodies can be produced. Okay, so the antigen is emulsified with FIA. This fuels in incomplete adjuvant, and this what does it consist of? It consists of a mineral oil and an emulsifier, such as manninite, monotheate. Okay, so these are the chemicals as I was mentioning. So these things are something that you do not need to just know what it is, but no need to memorize such things like mineral. If you have an adjuvant, it is having these chemicals, mineral oil and emulsifier, and it will form a stable emulsion that will allow the slow release of the antigen over time. Okay, it will slowly release the antigen, and as a result, it will prolong the exposure to the immune system. Your immune system will be triggered for a very prolonged period of time. And the oil component helps to create a depot effect at the injection site, promoting a stronger and more sustained immune response. So there are different adjuvants actually. By combining the antigen with Freon's incomplete adjuvant, researchers can effectively stimulate the production of antibodies and T cell responses, make it widely used technique in immunology. Okay. So the method used to determine the antibody titer, just now we discussed that. How will you determine the antibody titer? How much antibody is present in your blood sample? How will you determine? You will do an ELISA. You will do an ELISA. Okay, so in your ELISA you have 96 well plates, and in these plates you have the antigen immobilized on it. Okay, the antigens are immobilized on these wells, and then you add the blood sample or whatever sample you have. You will add this. Your sample will have antibody, so your antibody will go and bind to your antigen. Okay, so if your sample has less amount of antibody, then it will bind less. If your antibody the sample has more antibody, it will bind even more. Then you will use another antibody against this antigen, and this is the secondary antibody. This secondary antibody is conjugated to a fluorophore. Is conjugated to a fluorophore. So when you add the substrate, a colored product will be formed. Okay, you will get a colored product, and by measuring the intensity of the colored product, you can quantify that which sample has higher antibody. So in direct ELISA, you can read about this thing. The same thing is written. So you need to do washing, and then you will do this secondary antibody. So these are the steps. Now I will come to GST affinity chromatography. So what is gluta? So GST is basically okay. How many others close? Okay, so basically what is GST? GST is glutathione S transferase. It is an enzyme. Glutathione S transferase. So this is an enzyme which is present in the cells of our body, and this glutathione S transferase, this is an enzyme. So this is an enzyme, and what is the substrate of this enzyme? The substrate of this enzyme is reduced glutathione. Okay, reduced glutathione. So what is done that your protein which is present, your protein is tagged with GST tag. Okay, so GST is another enzyme, basically. It is another enzyme. So this is your GST tag and this is your recombinant protein. Okay, so what will happen when you apply this on the column? So you have your GST column. So this is your GST column. So if you see the GST column, then what will you get? So your beads is immobilized. Your beads is immobilized with glutathione substrate. So this is the glutathione substrate. So your beads are immobilized with glutathione. So once you add this lysate, so basically you lyse the cell. You lyse the cell, you have a bacterial lysate, and you pour this bacterial lysate inside this into this column. So what will happen? All the proteins which are having your recombinant protein, because your recombinant protein has GST tag to it, it will go and bind to your glutathione immobilized beads. Okay, you have the GST and you have the protein. So your GST tag protein will bind to your glutathione beads and all the other proteins. They will get washed off. Okay. Then what is done in the next step? In the next step, to elute the protein. To elute the protein, what is done? To elute the protein, you add increasing concentration of reduced glutathione. Okay, you will add increasing concentration of glutathione. So once you add increasing concentration of glutathione, what will happen? This protein, this recombinant protein, rather than forming or rather than being bound to this glutathione, immobilized glutathione, it will go and bind to the free glutathione. So, okay, so it will go and bind to the free glutathione, and it will no longer bind to your immobilized glutathione. So you need to increase the concentration of glutathione. So you need to add increasing gradient of glutathione to your column, increasing gradient of glutathione. So once you add increasing concentration of glutathione, your GST type protein will no longer bind to your immobilized glutathione. Rather what will it do? It will go and bind to your free glutathione and it will get eluted. Okay, that is the principle of a GST column purification. So see this figure. So you have a glutathione affinity matrix. So this matrix is immobilized with what? It is immobilized with reduced glutathione. Your GSH is reduced glutathione. So your matrix. So in this case, what is your receptor? Your receptor is a. Your receptor is what? Your receptor is a substrate. It is a substrate. And GST is what? GST is the enzyme. GST is the enzyme. GSH is the substrate. Okay, and this is your target protein. Your target protein is tagged with the GST tag. So it's fused with the GST tag. It's fused with the GST tag. So only your GST tagged protein will bind to your column. Others will get eluted. And then to elute your protein of interest, what you do, you add increasing concentration of reduced glutathione. Once you add increasing concentration of reduced glutathione, your GST tagged protein will no longer remain bound to the immobilized reduced glutathione. Rather, it will go and bind to your free reduced glutathione and it will get eluted. Okay, so you can read all this. So basically, if you want to recombinantly express and purify a GST tagged protein, you need to clone your protein in PGEX vectors. So you have PGEX vectors which are commercially available. So your PGEX vectors have they have this GST tag in their sequence. Okay, so if you clone the protein in this PGEX vector, then you're in the multiples after then yeah in the multiple cloning site. Post the multiple cloning site, there is this GST tag. So your protein will be expressed along with this GST tag. And another function of expressing a GST tag protein is that this GST is basically soluble. So it also helps in making the protein of interest soluble. Okay. So this is reduced glutathione. Reduced glutathione is a tripeptide. Remember this. It is a tripeptide, and it is why it is a tripeptide because it is made up of three amino acid residues. One is glutamic acid, one is cysteine, and another is glycine. So reduced glutathione means what? Reduced glutathione will have SH group. Okay, reduced glutathione will have SH group. Okay, and your oxidized glutathione will have this structure. This structure. Okay. So this reduced glutathione is the substrate of which enzyme? It is the substrate of GST. So GST specifically binds to reduced glutathione. It does not bind to oxidized glutathione. So remember this. So in the MCQ, you can be asked that GST, the substrate of GST is reduced glutathione, oxidized glutathione. Then the answer is reduced glutathione. Okay. So this is the figure which is taken from this website. So you can see here. So this is the bead and this bead is immobilized with this glutathione. Okay. Reduced glutathione and your protein, the recombinant protein which you have expressed that is fused to your GST tag. So this is your recombinant protein and it is fused to your GST tag. So what happens? Your GST tag, so GST tag protein will go and bind to your to the beads which is having this glutathione, which is having what? Which is having this immobilized glutathione on its surface. And then what you can do, you can your protein will get attached. Then actually there are different cleavage sequences. There is a thrombin sequence present in it. So there is a thrombin sequence present in it. So you do not want the GST. You only want this protein. You do not want GST to come along with your protein. So you can use a cleaving agent. You can use thrombin. So your thrombin, what will it do? the thrombin will cleave the GST from the protein and your protein will then get eluted. Okay, while your GST will remain bound to your column. Okay. You can read this as well. So you see here, purified fusion can be treated with the thrombin to remove the GST tag from the protein of interest. So you are basically have a matrix. Your matrix is immobilized with glutathione, and then you have the protein. Your protein has this GST tag along with it. So your this protein will go and bind to this glutathione, and then for eluting, you add increasing concentration of reduced glutathione. Okay. And when you will so your protein will also have the GST fused to it. So to remove the GST from the protein, you add thrombin. What do you add? You add thrombin. So your thrombin will cleave the GST from the protein and you will get the pure protein. Okay, the substrate utilized by GST fusion protein purification is what? It is reduced glutathione. Okay, glutathione is a tripeptide and it has three amino acids, glycine, cysteine, and glutamic acid. Your cysteine, when it is reduced, when it is existing as SH, that is reduced cysteine. That is when it is reduced glutathione. But if your when this thiol group is oxidized, then it will be oxidized glutathione. Okay. So this EST tag, it is a very large tag actually. This is a very large tag. It is having a size of 26 kilodalton. And so there can be when you're doing any biophysical or this protein experiment, you want to determine the structure of a protein, then you would not want this GST to interfere with the with your protein. Okay, because in presence of the GST, your protein can have a different conformation compared to when it is free. That is why what we do is we cleave the GST tag and then we go for further studies with the protein. Okay. So GST tag from the protein of interest is removed in GST-based purification by what? It is cleaved by thrombin. Okay, answer is thrombin. So basically there is a thrombin side. The vector, this PGX vector is designed in such a manner. So you have these different vectors, PET vector, PGX vector. We discussed all about all these, isn't it? So this PGX vector is designed in such a manner that that in between the protein and your GST sequence, you will have this thrombin sequence. Okay, there is thrombin sequence. So this thrombin sequence will be provides a cleaving site for action of the thrombin. Okay, so see this figure basically. So you see that this glutathione is transferase tract protein can be cleaved by thrombin. Why? Because this PGX vector series incorporates a specific thrombin recognition site. So there is a thrombin recognition site. This one you see here. This is this has a thrombin sequence site recognition site between the GST tag. So this is the GST tag. This is the protein. In between your GST tag and protein, you have your thrombin recognition site, and this thrombin so what is the recognition site of this thrombin? So it is a serine protease. Thrombin is a serine protease, and that is why it will recognize and cleave the consensus sequence leucine, valine, proline, arginine, glycine, serine. So it will cleave between the arginine and glycine. So we learned about these different fusion tags. How do we purify the different fusion tags? This in the last session I talked about these. So for thrombin, it will cleave between this arginine and glycine. So there are trypsin, there are thrombin, then there are cyanogen bromide, which will cleave after methionine. So this is how you can separate the protein from the GST tag. Now another way of so this is antibody purification. So how do you, so you have your matrix, and how do you couple the receptor or the antibody? Basically, how do you couple the antibody with your matrix? So for coupling the antibody with your matrix, you can go for this ENBR mediated receptor coupling. So this ENBR mediated receptor coupling is for antibody purification. So this is your polysaccharide matrix. So your polysaccharide matrix has have these free OH groups. Okay, and when you add CNBR, so CNBR is cyanogen bromide. This is a chemical. This chemical it will go and form will form this cyanate ester. So cyanogen bromide will react with this polysaccharide matrix having this free OH group, and it will form cyanate ester OCN, and then it will form this cyclic compound, cyclic imidocarbonate. And then once it has formed this cyclic imidocarbonate, then this receptor molecule which is having this free amine group, it can form this isourea derivative. So isourea derivative, you see that this structure has formed OCNH, and then this NH2 plus. So this is isocyanate, isourea derivative. So in this way you can, so what you have prepared, ultimately you have immobilized the receptor to your matrix. Okay. So what is found by this cyanate ester is formed when you treat, so these are chemical reactions. So when you treat it with cyanogen bromide, it will form this cyanate ester, and then this cyanate ester will react with the free amine group present on the receptor, and you get a isourea derivative. And this way your receptor remains immobilized on the surface of this polysaccharide matrix. So you can read all this CNBR, and it was covered in the lecture as well. It will react with polysaccharide at pH 11 to 12, and it will form reactive cyanate ester with matrix or less. Okay. So this is the reaction that takes place. You have this free OH groups which is present on the polysaccharide matrix. This will react with cyanogen bromide at pH 11. At pH 11, the cyanate ester is formed. Then the cyanate ester will form the cyclic compound, imidocarbonate. Then this imidocarbonate can form. So this ligand will have free amine group. This free amine group will react with this imidocarbonate, and you will get this receptor or this antibody immobilized on the matrix. Or what else can happen if so there will be some cyanate ester that will be formed, there will be some imidocarbonate that will be formed. If it is cyanate ester, then you will have this isourea derivative. You will have this isourea derivative. So there can be this one also forming, or there can be this also forming. So you can have either isourea derivative, or you can have this imidocarbonate forming this complex. Okay. So basically, cyanogen bromide activation is one of the most widely used and well-established methods for coupling ligands to solid supports in bioaffinity chromatography. So how can you use cyanogen bromide for coupling your antibody to your matrix? That is what is explained here. And this technique allows for the covalent immobilization of receptors, antibodies, enzymes, other biomolecules. So if you want to covalently immobilize your receptor or your antibody or your enzyme to your matrix, then you can use this cyanogen bromide. It will react with the hydroxyl group present on agarose matrices. So that is what in the figure I was showing you. So there is the agarose matrices will have the free hydroxyl group, and under alkaline conditions, what form? It can form two intermediates. Either it can form cyanate esters or it can form imidocarbonates. So this one, either it can form this intermediate or it can form this intermediate, imidocarbonate. Okay. The activated matrix will then react with the primary amine group present in proteins and other biomolecules. So if cyanate ester reacts with the free amine group present on the receptor, then it will give an isourea derivative. But if it reacts with the free if this imidocarbonate reacts with the free amine group, then it will give a substituted imidocarbonate. Your isourea derivative is positively charged, but your substituted imidocarbonate is neutral. Okay, this is neutral, while this is positively charged. So it can act as a cation exchange chromatography column. Okay, it is positively charged, so it will be anion exchange basically. Okay, it will bind to negative charges. So the type of group does CNBR form when reacting with a polysaccharide matrix at pH 11 to 12. So just now we said that this has matrix and this has free OH groups, and then this cyanogen bromide will react with this free OH group, and it will form what? It will form OCN. This type of molecule is formed, and this molecule is what is called? It is called a cyanate ester. It is called a cyanate ester. OCN. Okay, cyanate ester. So it forms cyanate ester groups, and in this alkaline environment, cyanogen bromide react with hydroxyl group present on polysaccharide that will lead to the formation of cyanate ester through a substitution reaction. The modification introduces reductive cyanate ester. You can read about all this. Okay, same thing is written here. So basically, you can do a nickel NTA column. This pull down assay also you can use using nickel NTA pull down, or you can use a GST pull down. So that is what is explained here. Matrix is incubated with pure protein 1 and then washed to ensure tight binding. So if you want to study that whether protein 1 interacts with protein 2, how will you do that? If you want to study if protein 1 interacts with protein 2, then you can do this. Either you can do a GST pull down or you can do a nickel NTA pull down. So matrix is incubated with pure protein 1, washed to ensure tight binding. All other sites on the bead is blocked with a non-specific protein such as BSA. So you add BSA so that there is no non-specific binding. And now the cell lysate of the pure protein 2 is passed. So first you immobilize the protein 1 on the matrix, and then so your protein, you have purified the protein. For example, it is a nickel NTA or it is a histide protein. You have immobilized it on the matrix, and then you want to see which protein does this protein 1 interact with. So you add a cell lysate. Your cell lysate will have different proteins. So the one will have different proteins. The one which is having affinity for protein 1, that will only remain bound to the column. The rest will get washed off. So you do couple of washing steps, and after the washing steps, you add the elute. You add the elution buffer. Once you add the elution buffer, if your protein 1 is interacting with protein 2, then you will then both of them together will get eluted. But if it does not interact, then what will happen? Your protein 2 will get eluted separately, your protein 1 will get eluted separately. And how will you understand if your protein 1 interacts with protein 2? You will run an SDS page. So if you run an SDS page and if you see that that fraction, then the elution fraction, you have bands of both protein 1 and protein 2. It means that protein 1 interacts with protein 2, but if it so happens that in your wash fraction, your protein 2 gets eluted, then it means that protein 2 does not interact with your protein 1, and you will get separate band of protein 1 in one lane and separate band of protein 2 in the other lane. Okay, so this is a J pull down assay, which is done regularly to study the interaction of one protein with the other protein. Okay. So immunopurification is what? So avidine biotin system is used to capture and isolate cytokines from the immune cells. So biotinylation of antibodies allow immobilization of the antibodies in the correct orientation. So basically here what you are doing, your antibody is biotinylated, and then your biotinylated antibody is what? It is immobilized. It is immobilized on the matrix, and then your lymphocyte lysate is passed to the column packed with the glass bead containing antibodies. So this is your lysate lymphocyte. So you have your lymphocyte cell. You have your lymphocyte cell. You have lysed the lymphocyte cell, and then you have lysate. You will get a lysate. Okay, and then you add the lysate to your column. You will add the lysate to your column. Your column has what? It has immobilized antibody. Okay, so if your lysate has the cytokines which has affinity for the antibody, they will bind, other will not bind. In that way, you can identify which cytokine has antibody for which type of, or which is that cytokine which is having affinity for your target antibody. Okay. By following or decreasing pH or by cryotropic agents, ions, and the antibody remains bound to the column due to strong affinity, which is resistant to chemical treatment. So this is immunopurification. Okay. So the last two questions for this session. So why are you using BSA? So the purpose of using BSA was what? So the purpose of using BSA is to block non-specific binding. Is to block non-specific binding. Okay. So when we are adding BSA, we are adding BSA in Western blot as well. In this also pull down assay, also we add BSA so that there is no non-specific binding, so that your antibody does not go and bind to anywhere in the column. It will go and specifically bind to the antigen, or your antigen goes and specifically binds to the antibody immobilized on the resin. It does not go and bind anywhere on the say for example glass surface or on the anywhere on the beads. Okay, block non-specific binding. Okay. So why do we use biotin-islated antibodies in immunopurification? The answer is that it provides correct orientation on the beads. Okay. So why? So advantage of using biotin-islated antibody in immunopurification lies in their ability to provide correct orientation when bound to streptavidin coated beads. So your beads are coated with streptavidin and your antibody has biotin. So biotin streptavidin interaction. So biotin has affinity for streptavidin. So it will your antibody will go and bind to your matrix, which is immobilized with peptidine. Now, this strong interaction ensures that antibodies are oriented properly, maximizing their accessibility and binding capacity for the target antigen. Okay, so by maintaining the correct orientation, it is essential that your antibody maintains correct orientation. Otherwise, it will not be able to bind to your antigen. That is why we use biotin-islated antibody. Why? Why do we use biotin-islated antibody? So that it can maintain correct orientation when it will bind to the matrix which is immobilized with streptavidin. Okay. So that is all for today. So I will stop today's session now. So if you have any more doubts, you can ask me. Next we will see in the next class. Okay. Thank you.